Alright, so today's class we're going to dive a little more deeper into how evolution is actually enacted. So we looked at adaptations and variations, but however we don't know how specifically are they developed. So today we're going to be looking at those. So we're looking at the mechanisms of evolution. We're looking at the entire process. We have mutations, which is how every single adaptation or variation starts. We're looking at gene flow. We're looking at, well, so how genes change in a period of time. We're looking at non-random mating, which means that people, they are mating based on looks or based on ability to find, prey or whatever. And then we're looking at genetic drift, how genes could be driven, how the gene trend could be driven. We're also looking at natural selection. And again, we're looking at microevolution, which is the frequency of an allele change within a population. So, number one is mutation. Well, how is mutation given? Well, it's a change in DNA, so it provides new alleles that produces variation. So an example of this is poison resistance of the Norway rat. So Norway, they used to ship stuff around. And obviously when you ship stuff around, there's going to be stuff on the ship that's going to be, attractive to the rats and, all of these mice. So they came on the boat. And obviously they're now eating the stuff. Like let's say green tea, they're eating the tea. So obviously when you eat the tea, you lose profit. So they came up with a genius idea of trying to poison all of the rats. So suddenly one of these rats, they had these mutations that allowed them to now be resistant to this poison, to this rat poison that they gave them. So now suddenly that boat is full of a boatload of poison resistant rat, and they couldn't do anything about it. So they came up with the idea that, oh, maybe mutations, they brought us some sort of resistance to these rats. So now we gotta up the poison. So in warfarin is a thing used in rat poison. It's used in very small doses, so it takes around 6 days for them to die. So it was originally from Asia. It was called Norway rat because it made appearances in these ships. So mutations made some of them resistant to warfarin. And if you know about rats, they have extreme high reproductive rate. So these things, when they got this one rat, they became resistant to the poison. Suddenly, the entire population is now resistant because of how fast they reproduce. So we gotta know that mutations is completely random. Some and very few amount of them is actually beneficial. And a lot of these mutations that eventually reach high frequency in populations is usually neutral. So the majority of stuff, the majority of mutations that we have is actually just pure neutral. They don't do anything. This, however, a neutral thing. So, a neutral thing that was once neutral in a period, it could be beneficial when the environment changed. But however, these mutations, they usually just go around quietly around the population because we don't even notice. So neutral mutations, remember, we don't notice them. They just don't do anything. They don't hurt us. They don't do anything, So, however, there were genes that were detrimental and harmful, but so they didn't survive. So that's why we don't have them nowadays because the ones that were detrimental, it obviously killed them, and now they don't have them anymore. So we're going to look at the idea of gene flow. The transfer of allele from one population to another. Well, how does gene flow happen exactly? It could be because somebody moved. Let's say I moved from Asia to, let's say, North America, and I had this. Let's say I had a bad allele. So now okay, let's not use humans. So there was this dog that came from, let's say, Asia. It had a bad allele. Now it reproduces in North America. It's going to produce people or dogs that have these bad alleles. So individuals that transfer in or out of a population may be responsible for a change in frequency. And individuals moving in may bring new alleles. So I want you to understand that in a very small population, let's say only of three dogs, if two of them had a good gene and one of them had a bad gene, so now you have 66% of good genes and 33% of bad genes. But suddenly, when this bad gene, that one dog left, you're back to 100% good genes. So if a population is very small, this change is really, really big. And that's the kind of the idea of gene flow. And you see, these are how gene flow changes. The left, it's actually pollinated by a bumblebee. So um, when we have cross-pollination, eventually the color is going to change, the shape is going to change, and all of that, everything's going to change. On the right, it was pollinated by a mockingbird, I believe. And also you can see the color change and how its shape completely changed. It's so very interesting because of gene flow. So the idea of non-random mating comes into play whenever we step into the real world. So when individuals select mates based on their phenotypes, and what I mean by phenotypes, it's things you can observe, the actual stuff. So inbreeding. Inbreeding decreases the proportion of heterozygotes, and it increases the chance of receiving recessive abnormalities. So some of the genes are dominant, some of them are recessive. That we know that, we should know that, and we're gonna go deeper in genetics. So when we have people that keeps on inbreeding, so let's say we had a heterozygote mate with a homozygote positive, and now they're going to have a great chance of getting a heterozygote. And essentially, when we inbreed, let's say a dog, we breed it with its close relative, it's going to have some sort of weird genetic diseases, because that's why humans don't inbreed. That's why it's not allowed in laws. So when you see self-fertilization of flowers, you see that these flowers eventually they have all of these problems. They might be more vulnerable to diseases, they might be less colorful, or they might be less attractive to pollinators. So we're gonna look at the idea of black widowbird. So non-random mating, remember, we have their tails essentially. So some human went in there, we cut these tails. We cut their tails, so now their tails is shortened. And we put this shortened tail, we cut them down, and we put them onto other widowbirds. And now suddenly other widowbirds, they have longer tails now. So you see, this is a number of mates. So these super long tails, they tend to get almost 2 mates. And these they don't even, and these shortened ones that we cut it off, they don't have any mate. They barely get 1. And these normal ones, they get around 1. So this is completely not random mating, because not all of them have the equal chance to mate. And this one with the huge long tail, it clearly has a better chance to mate than others. Genetic drift. We're going to talk about the idea that there could be changes to allele frequency completely due to a random event. So there's 2 effects that we learn. It's the founder effect and the bottleneck effect. So founder effect, let's say a couple of people, Europeans, so they founded a colony all across in North America. And now so North Americans, they do have some sort of weird diseases. So let's say we had 5 Europeans that each of them, they were heterozygous, meaning that their recessive disease, they don't show, but they're a carrier. So when they come to this new land and they reproduce, the chance of the homozygote recessive, the chance of them getting a weird recessive disease is going to increase gigantically. And that's shown by here, the population of the Amish population and polydactylism. If you don't know, polydactylism is you have this weird 6th finger, I believe. So Amish population, they don't like, they had all of this weird recessive diseases. However, they want to maintain their bloodline, so they kept on reproducing within the population. And then obviously this weird recessive disease is going to be spread all around. And then now suddenly, whenever you see an Amish, they're like, "Oh, can I see your finger? Oh, it's polydactylism." Well, and another thing that we should look at is that English, I'm pretty sure the Britain monarch and the Japanese monarch, they do this too. They want to maintain their bloodline clean, and they want to be like, "Oh, this is an imperial bloodline. It's superior than normal people. So we're gonna inbreed with our close relatives to maintain the bloodline." So suddenly all of them, they have this weird diseases. I'm pretty sure for the Britain, it's okay. I'm pretty sure they have like hemophilia or something. But for the Japanese, they're rather short and their IQ is highly lowered, like much lower. So the other thing is bottleneck effect. sometimes a population becomes almost extinct because of natural disasters. So we had this. Let's say we have like an example here. So there was 1775 people on this island, and there was only 30. Oh wait, this is a year. Okay, so there were 16,000 people, and only 30 of them survived. We don't know what these 30 people have. These 30 people potentially they could have had like a weird genetic disease, but they didn't show it. So okay, I can't look. It's right here on pretty close to Russia. I believe this is China, this is South Korea. Yeah, this is on part of Russia, I think. So 18th century, there was a large storm that went over the island and killed almost everybody but 30 people. So almost 4 generations after this huge typhoon, the citizens of Pinglap began to exhibit symptoms of a very, very, very rare recessive disease named achromatopsia. So it's characterized by extreme light sensitivity, poor vision, and complete inability to distinguish color. So roughly 30,000 people living on the island today. 5 to 10 of them are affected by this disorder, and around 30 of them are carriers. So what does that mean? Maybe around 5 or 6 people out of this 30 people that survived, they had this disease. And because they were the only ones there, and they were the only ones that's able to reproduce, they passed this disease on. And essentially, all of these people could all of these people are probably able to trace their ancestry to this one single male typhoon survivor that reproduced and had a lot of kids, had a lot of grandchildren, who researchers to believe was the carrier of the diseases. And because his descendant, most of them, well, they probably were carriers, but because they intermarried and they inbred, they had these kids that were closely related. Now they have an increased chance of getting this disease. So today's a short class, but I want you to know that because of us, we kind of lowered a lot of genetic variation, and we put some animals to some pretty dangerous states. So we hunted a lot of these kids, and we ate them because they were delicious. So it was a very, very little. We killed almost everyone except for like 20 individuals. So now we humans, we suddenly became aware. We're like, oh no, it's going to die. We need to revive it. So it came back to around 30,000 population. However, there was only 20 to start with. So these 30,000 population of seal or whatever that is, it all came from these 20 individuals. So that means that they are probably carrying some sort of genetic disease, because they have to inbreed. Only 20 people, you're not going to grow the big population without inbreeding. Cheetahs. So cheetahs, they were actually endangered at one time because of habitat loss and, well, hunting. So cheetahs, they also have a small gene pool because of us. They have an abnormal sperm, and they have very bad immune system, and they have these viruses that can kill them. So now we're kind of looking at linking over the world. So United Nations, they have a call to action for this. So you have something in China, they have this cheetah, and then they're shouting out or going around to post their media posts on United Nations, and they're looking at the least related cheetah to my cheetah, and they provide the DNA sequence and all that. So now they're trying to get the least related ones to reproduce. So now that they increase some sort of genetic diversity, but they aren't doing so great. Yeah, so the big takeaway of this class is just how has evolution changed, and you just gotta look at these 5 things. Remember, they're all microevolution because they're looking at the frequency of an allele change, and microevolution is like creation of an entire species, and we'll look at that in a couple classes.